Okay, hi everyone. Welcome to our February SSCD webinar. This webinar is sponsored by Takeda, and we thank them for their generous support. We do have CME and CEU approval for this webinar, so I'm going to share my screen and we'll share it again at the end so everyone can learn about the process for claiming their credits, but we are receiving credits through Boston Children's Hospital. And so you've got to go through their process in order to claim the credits. So you've got to set up an account with them. I will share the link to this website, but it's dme.childrenshospital.org/account registration. And I will share that link so that you can get to it easily in a minute. And then once you sign up, if you already have a profile, you don't need to do anything. You can just text this code, this 459242 to this phone number here. But if you do need to create an account, if you have not claimed credit before, you'll need to log in, create your account, and then text the code. Make sure your phone number is associated with the account so that it will log you appropriately. So again, we'll share this all in the chat. If you have questions about it, you can certainly contact us after the webinar. I will make sure to get it to everyone afterwards. All right, so we have two wonderful speakers today. We have Victoria Kenyon, who is a clinical research project manager at Mass General Hospital for Children, and Dr. Sunita Krishnareddy from the Celiac Disease Center at Columbia University. And they will be talking about best practices in clinical trial design. So I'll hand it over to you both.
You. Um, Thanks, Sarah. Okay, everyone could see our screen clearly?
Yes.
Okay, perfect. So we are, sorry, that went all the way to the end there. We are both going to be talking about kind of the different types of trials in celiac disease, the benefits, the pitfalls, the difficulties, all that kind of stuff from both of our points of view. So I'm going to start a little bit more on the overall what trials exist, why they exist, what has been helpful and hurtful in trials in the past, and then I will hand it over to Victoria, who's going to get through the more nitty-gritty details of how to actually run these trials at a center. These are our disclosures and getting right into it. So those of you who see patients, interest in new therapies is actually relatively high. So I remember being at a celiac meeting probably in 2015, right when we were starting to talk about this a little bit more in these big general settings, and asking patients if they would be interested if a medication ever came about. And surprisingly, many people said no. You know, they were not clear on the risks that would be associated with this. They thought, if a diet is working, why do I want to take a medication? Many of them are parents, and I'm adult gastroenterologist, so I don't see this firsthand, but I do know in speaking to the parents, obviously this is a big deal, right? To put your child on a medication for something that maybe you're managing totally fine with diet alone. However, and because the diet is so effective, this has been the mainstay of treatment and patients have gotten used to it. However, we're seeing more and more that the diet is challenging, right? It's expensive. Socially, it can be kind of isolating, especially for children when they can't eat the foods that their friends are eating, go to birthday parties, have cake, stuff like that. But also, even those who are very strict with the diet, there are still some people who just have persistent symptoms, and even very small amounts of gluten cause these symptoms. And it's if for anybody who's ever been on a diet, you know how careful can you be. So this is something that comes up a lot now. Now we see when we talk to patients, a lot of what they want is protection from inadvertent gluten exposures. It's not even like people want to go and just eat a whole big pizza every single day. It's just when you're traveling or when you're at a work meeting or when you're in a place where you're not controlling the situation, how do you get over those gluten exposures? And then also the nervousness of what happens when you're getting these long-term gluten exposures, and if those cause tissue damage and if they're causing other issues. So patients are worried about that as well. I'm sorry if the noise is a lot in the back. I'll try to be louder over it. But the different categories of medications in celiac disease are a few. So some are detoxifying gluten prior to its presentation to the immune system in the small intestine. Some are immune modulation, so really actually decreasing our own immune responses to these immunogenic peptides. Some are tolerance promotion, so even if you do get exposed to these gluten peptides, are you going to react the same way if we kind of promote tolerance in those patients? So those are the main categories. Are we close to these medications? And, every year patients, I notice whenever I talk to them, I feel like I'm doing this too. And I keep telling patients, maybe in 5 years, we'll have something available for general use. But the target keeps moving because we've learned a lot from the way that celiac trials are designed. And up until recently, we really didn't have a good standard for that. So I actually do think now that this has become a little bit more generalized over how the trials are put in effect. We will be getting closer to some of these medications actually making it through this very rigorous process. So the clinical trial phase, I'm not going to go into it too much, but it is quite rigorous, right? So phase 1 is the safety phase where we're just making sure that medications don't cause some horrible side effects. About 60 to 70% of medications make it from phase 1 to phase 2. So it's relatively high. Phase 2 is where the nitty gritty comes in and you're actually looking for, is there an effect of this medication? So in addition to more safety data, we're trying to see what is the appropriate dose, how these medications work, are they actually doing what they're supposed to be doing? And the movement of phase 2 to phase 3 is actually the smaller movement. So about 30% or a third of these drugs get moved on to phase 3. So they find good results and they can move on. Phase 3 is kind of what we all see when we see trial results, right? These are the ones that are published in JAMA and that make news and whatnot, because that's the closest to them getting approved by the FDA. They're usually very large studies, so they require hundreds to thousands of patients to be involved. We're looking at safety again. We're usually looking at it compared to a standard therapy like the diet or a placebo if we're looking at mitigation of damage. Once it goes through phase 3, that's when it then goes to the FDA for approval. About 40 to 50% of those medications that have positive phase 3 actually get approved by the FDA. So it's not a slam dunk just because it gets through phase 3 that it's going to be approved. And then there's phase 4, which we don't really hear about, but these are like the long-term maintenance trials that you see when drug reps come and visit you or when you're looking up data for other medications, the ones that show you, yes, not only does it work in the short term, but it actually works in the long term, and these are really good medications. In celiac disease, a couple of the issues that we've had in the past with trials, and I'll go through specific trials later, but it's looking at key clinical endpoints. So what are we trying to improve for with these medications? In the past, it has been very symptom heavy. So looking at the celiac disease symptom diaries or patient reported outcomes, the celiac symptom index or celiac disease questionnaire. Any of you who have ever done that for any patients know that these are very not specific to celiac, right? So a complaint about diarrhea, abdominal pain, constipation, those can happen even when patients are not eating gluten when they're celiac. So how do we know that gluten is what's causing these issues, right? Later on, the trial started including more histologic endpoints. So we're looking at villus height to crypt depth ratio. That's the most common primary endpoint now because it is a more objective measure. It does require biopsies, which is obviously more invasive for the patients at baseline and at some point during the trial, but it gives us a way to standardize what we're looking for because usually these biopsies are read by a standard panel. They're measuring objective findings on a biopsy to say, okay, this person has improved their ratio and now it looks like their damage is a little bit less. And that's maybe a little bit more quantifiable. Also, sometimes looking at intraepithelial lymphocyte density, that's also just an improvement, not an absolute cutoff, because those numbers maybe never improve even in patients who feel 100% better on a gluten-free diet. So these are all taken as ratios from beginning to end, not absolute values. Another thing we can look at is serology improvement. So if patients have persistently elevated TTGs, and with the new treatments, do those numbers go down? And is that a surrogate for then improvement in histological healing? And then also gluten peptides in the urine, which we use to actually help patients who are eating out or traveling and trying to figure out what foods might be triggering them. But do we see decrease in that with any of these medications? Those are not usually primary or secondary endpoints in most of the trials, but they are things that we can use as surrogates. How do we choose patients? This is the tricky part, and this is where we come in, but how is the disease initially diagnosed? So if a patient is diagnosed because their family members had celiac and they were screened, or they had a concurrent autoimmune condition like diabetes or hypothyroidism, but they have no GI symptoms, is that the patient that you want to put in a study? Probably not, right? Because it's going to be hard to follow their symptoms when you're giving them the gluten and see if the medication is making any difference. Adherence to the diet is really important as well. If patients are not adherent at baseline, how are you going to see if a medication, which is supposed to be given in conjunction with the diet, is helping them more? It is good for real world data because most patients are not, there's variable level of adherence and it can actually change over time. But for a trial, you're probably not setting yourself up for the best endpoints if you're not getting patients who are very adherent to begin with. Persistent symptoms are also, a lot of the trials in the past have required patients to have persistent symptoms, but what are those symptoms? If patients have nausea, vomiting, diarrhea as soon as they eat gluten, that's probably a really good patient to put in a trial in the sense that you're going to know if the medication is mitigating those effects. It's not a good patient to put in the trial in the sense like, do you want to give that patient symptoms knowing they're going to have these very violent GI symptoms because you have to give them a gluten challenge? And that's something, again, we'll talk about a little bit later. Are the symptoms really related to gluten? Do you have a patient who says every day they feel horrible, or only when I go out and eat and I'm not sure if the restaurant was safe, do I feel horrible? So that's also a way to kind of figure it out. Age, most of the studies are just for adults, so that is a big component. But even within that adult age frame, are you choosing 20-year-olds who are living at college and maybe don't have as much control over their diet versus a 35-year-old who has like honed this to perfection over 10 years of having the disease and knows exactly what they should and should not be eating? And then other autoimmune diseases might just confound results a little bit. Some trials absolutely exclude patients with other autoimmune diseases, but that's really trial specific. Study design, and this is just general, but randomized control trials, longitudinal trials like they do in Colorado, which they do a fantastic job in, gluten challenge studies, animal models, and then retrospective case control studies. So we've done all of these in celiac. All of these exist. Which ones are the best? It depends on the question that you're trying to ask, right? So if you're looking for a new medication, you're not going to do a retrospective trial, obviously, right? You have to do this randomized control trial. But if you're looking for environmental triggers that are making symptoms worse, maybe one of these longitudinal studies or a case control study is something that can help you. So it's really just dependent on what you're looking for. The main trial designs for drug therapies are two. One is a gluten challenge study. So these are patients who are adherent to their gluten-free diet. You purposely give them some known amount of gluten along with a drug or a placebo, and then you see how that drug or placebo mitigates these symptoms associated with that gluten ingestion. You follow those symptoms and the histology. So these are for patients who are feeling pretty well, but willing to eat gluten under controlled circumstances. I don't know how many of you have asked patients for trials like this, but many patients, when you tell them there's a gluten challenge component of this, are not willing to do this. And Victoria will probably talk about that a little bit later on, but that is a hard sell, right? This is somebody who's feeling really well and working and going to school and whatnot, and you're telling them, I'm going to give you something that I know makes you sick, but hopefully the medication will make that better. You don't even know if you're getting the medication or not. So it can be very difficult. Recruitment is difficult in these settings. Dropouts, of course, if a patient realizes they're getting placebo and they keep getting sick, they're not going to want to stay in the trial. And then also, it might work in a trial, but that's a certain amount of gluten we're giving in a specific formulation. Maybe that's not what the patient eats normally. And in real life, this medication is not going to work because it's not simulating what real life gluten exposures are like. And then the concept of how much gluten and what formulation for how long, all of that has up until now been a little less standardized. There are the ethical considerations of a gluten challenge, right? So patients are making this decision on their own. These are adult patients We're telling them what the trial involves and they are choosing. We're not forcing anybody to be in this. So there is that component that says, okay, you are choosing if you're willing to do this or not. But as physicians, when we're recruiting patients for these trials, our main goal is to not hurt our patients, right? And if we're purposefully giving them something that we know hurts them, how do we reconcile that with science and advancement of science, right? Also, the whole concept of placebo-controlled trials, we're not telling a patient, this is not like a cancer therapy where you're stage 4 and you're definitely going to get treatment and we just want to see if it works. This is, we might give you nothing and then give you the thing that makes you sick, and we want to compare it to the people who are getting something, which is hard for us to reconcile as well. And then also, do those patients have very long-term complications with gluten exposure? So that goes back to recruitment, and maybe those are patients you should not put in a trial, because if somebody feels sick for a week post a gluten ingestion, that is not going to be a good participant for the trial, because you're going to take away so much quality of life from them during the trial period. And again, these are usually young, working individuals going to school, like very busy lives. They don't have time to be getting sick for a week at a time. So maybe getting a medication or not. Also, there is the concept that this celiac disease is relatively well controlled with a very safe therapy, right? So patients usually do well on the gluten-free diet. Are we now saying, let's take that away from them and give them gluten and then see if a drug might or might not work as well as the diet? Again, this is not like inflammatory bowel disease or cancer where their disease is progressing no matter what you do. This is a patient, for the most part, who is feeling well, and you're just trying to see if something is going to work. So the benefits in that case might not help with this. The long-term effects also, the gluten challenges come into play. So we, again, know we're giving them something harmful. It's like putting a patient in a diabetes medication trial and telling them to eat candy every single day. That is not how their trials work at all, right? So this is a kind of an unusual design for a clinical trial. It has become a little bit better with the siege type trials or the simulated inadvertent gluten exposure, because the amounts are a little bit more controlled. Instead of telling a patient, go home and eat 1 or 2 slices of bread and see how you feel. You know, that patient could mean, okay, I'm going to go home and eat 2 very huge slices of very crusty French bread, or like 2 very thin slices of like Ezekiel bread or something. So it's not controlled, and doing that makes it much harder to get good. Biomarkers are helpful in the sense that maybe the amount of gluten a patient needs can be a little bit less before we see rises in their cytokine levels, specifically IL-2. But that is very research-based, and that's not something we use in clinical practice. But in these settings can be helpful to kind of lessen the gluten exposure, time, and amount. Okay, the second type of trial, which we call like a real world trial, is you let patients do what they're normally doing. You give them the drugs and then see if their normal day-to-day is different with the drug versus placebo. So we assume some patients get some amount of inadvertent gluten exposures, right? Especially if they eat out, or even regular food that they prepare at home could have small amounts of gluten in it. That these trials are looking at, is that small amount mitigated with these medications? There is a higher risk of type 2 errors in these trials, because obviously you're not telling patients to eat gluten, which induces the damage. So maybe they don't eat gluten at all in a normal period. And then you have this participant who has no difference in drug versus placebo because they're not getting the trigger anyway. Also, on the flip side, maybe their symptoms are not related to gluten at all. So they're taking the drug, but none of their symptoms are better, but it's not because the gluten is not being adjusted with the medication. It's because some other food is causing these symptoms, or some other underlying illness is causing these symptoms. So there is a lot more room for error in these types of trials. And then there's a bias that happens when patients are in a trial, that they're always on their best behavior. They're not eating gluten, they're not eating out, they're being super strict because they want to look like the perfect patient, right? But that's not real life. Once 6 months are done and the trial's over, and they go back to how they were before, symptoms might come back then because now they're liberalized and we're just not catching that during the trial. The good news is, again, when I started GI fellowship in 2010, I don't think there was any treatment at all for celiac disease besides the gluten-free diet. Since then, it's really accelerated. The interest has accelerated from pharmaceutical companies, from patients, as we mentioned, from physicians wanting different options for their patients. But it has become a whole new world, which is very exciting for us as practitioners to share with our patients all these different medications, different mechanisms of action. And I'm not going to go through all of that because that's not what this is for, but just to show you guys what has happened in the past 10 years and what is happening. So these medications are the ones that are coming out in the next year or two in terms of trial results, sorry, not the medications themselves, but just showing there's a lot of development in this area and it's very exciting for us. What have we learned from the stuff that didn't work? So lorazepam, which is probably the biggest one because it's the first drug to get to phase 3 for celiac disease. It was stopped not because the medication didn't work, but because the number to show a difference was just too high. So what does that mean? First of all, the endpoint in that trial was clinical symptoms. So maybe that initial endpoint was not the best one because you're asking patients how they feel when, as I mentioned, maybe gluten is not what's causing them to feel bad in the first place. So when you do a clinical endpoint like that, then you do an interim analysis showing not enough patients in the test group are showing improvement, but we don't know why. And then you stop the trial because the N becomes too high, you might have actually lost some response rate because actually maybe those patients had completely healed villi, but we didn't know because we were not biopsying them. What we know from previous studies is even though a drug might fail a study like that initially, maybe looking back at the subgroup analysis gives us some data that we can use moving forward and say, okay, well, maybe this medication actually worked in this specific group of patients. And maybe we should just study that one group because the N is not going to be so high and we might see the specific results for that group that are positive. So that's what we're waiting to see with lorazepam. With NextVax, which was a vaccine looking at gluten tolerance or immunotolerance of gluten peptides, maybe what we learned is that it's not easy to induce tolerance once you're already intolerant. That is a component of our immune system that is very hard to control. But also that celiac disease is kind of multifactorial, and it's not just tolerance to these peptides that prevent you from getting symptoms, but something else as well, right? IL-15 also failed its initial trial looking at refractory celiac disease. We know in general refractory disease is very hard to classify. The number of patients is very low with refractory type 2, which was specific for this trial. So even though that trial failed, they actually repurposed the medication and are looking at it for a different indication, which is a great way to kind of move these drugs through, right? And that's what I said with lorazepam. Maybe it doesn't work for everybody with celiac, but this specific population will benefit from this medication. And then what we have learned is the treatment we have, the gluten-free diet, is very good. So it is an excellent treatment. It sets a very high bar because you're getting symptom resolution in most patients. So when you're trying to show that something is worth testing a medication, you have to show that it's at least as good or better than the known treatment. And that becomes very hard when the known treatment is so good. So what should we be looking at instead? So these are just some key takeaways that the immune system is sensitive. One mechanism is probably not responsible for celiac disease, as we know, right? So just stopping one thing from happening doesn't necessarily stop the symptoms that come with celiac. Barrier modulation, like in lorazepam alone, was not sufficient because there's some other component turning on the celiac. And I already mentioned inducing tolerance is very difficult once you're intolerant. And that the trial design is just hard because these are patients who you're giving a known trigger to, who you're comparing against a treatment, which we know is extremely efficacious. So it is a harder bar than actually most. And celiac disease is more complicated than we thought. Challenges that we've seen thus far in drug development related to that is, again, our symptom endpoints. So what are we looking at? Biomarker endpoints, how do we standardize this? And then what is an acceptable amount of gluten that we can be giving patients? Luckily, there was a FDA group that met together to kind of decide what are the important components of this and what should we be looking at. And they came up with this. So they want to standardize trial design, eligibility criteria, and the gluten challenge methodology. But clinical trials with the gluten challenge should not include any FODMAP foods that got cut off. And that clinical trials should be treating celiac disease symptoms, but how are we going to measure that? So patients in a gluten challenge type trial need to have been on the diet for 12 months or more. This is now FDA guidance as to how these trials should be run. And that their baseline endoscopy should be relatively low. So they should not have persistent bilicentin. The challenges, like I said, should be free of FODMAPs foods because we know that those also induce a lot of symptoms, especially in celiac patients. And that using histology as a primary endpoint with a clinical outcome as a secondary endpoint is the best way to measure these trials. Because again, it's a little bit more objective on the initial aspect of the histology, but what we're really trying to treat is not a biopsy, but a patient's symptoms. So we have to correlate this with the patient-reported outcome. And then it gave some guidance as to what we're looking for in those histological endpoints, the villus height to crib depth ratio, and then in terms of clinical symptoms, it's a little bit more up in the air. The trial design itself can choose what clinical endpoints it's for. So what do we need to get a treatment? The sponsors, you need drug companies to be interested in this and to look into why there's a need for this in the first place. Consensus on those endpoints, which I think we're getting a little bit better at. So what is an effective treatment? Is it somebody who can now eat pizza with not having diarrhea? Or is it somebody whose tissues shows no damage after eating like a big gluten and containing meal? And then patients need to be okay with it, right? Are they willing to eat the gluten? Are they willing to take medications? What are the side effects that they're willing to tolerate for these kind of treatments? And then investigators like ourselves, we need to be looking for these patients, right? Screening our patient population and saying, oh, this is a good patient for this trial. And these are people who have symptoms that we can maybe treat in a better way. And connecting all those dots together, which is where our clinical trials offices really help with that, obviously. Now, let's say we're all in a good mood. We finally have a drug that works. We've done it. We figured out how to run a successful celiac disease drug trial. But now what happens? Who gets these medications? Who prescribes it? Can it be prescribed by a primary care? Are they going to extend it to children? Is something that needs to be taken every single day, or is it on and off? Do we tell patients take the medication and don't worry about the diet? Are these results going to hold up? All these are questions that are going to come up, of course. We will be lucky to have these questions in a few years, and I hope we get to that point where we have a plethora of medications to choose from, and our biggest concern is who gets it and how, as opposed to right now where we're still in the development phase. It's still in the kind of discovery phase. But eventually we will get there, and these are all things that we need to think about, and these are things that we need to think about when designing the trial, because those parameters that are put into a trial are what will help us eventually figure out how to properly prescribe these medications. So that's it for my part. I think we're going to take questions at the end. I'm going to hand it over to Victoria.
Perfect. So I'm going to kind of touch more on the operational execution and trial management from a site perspective. Obviously, none of these treatments or therapy options would be made possible if clinical trials are not designed properly or executed at the site. So it kind of starts from there. Next slide. So we'll kind of jump right into some recruitment strategies. And I will say that a lot of this information that I'm going to share are things that have worked specifically for my site or things that I've talked about with other sites just in my experience. So I always say to utilize both internal and external recruitment strategies. Internal, going off of your own celiac clinic, your own patient lists, whether that's the PI or any co-I on your trials, but also expanding that to other GI clinics, other GI providers, making sure that those GI groups are aware of a clinical trial that's going on in your site, so that if they do come across a patient in clinic or they remember a patient, they're able to refer that right to you. And also not forgetting about dietitian groups. They see patients pretty frequently, so being able to have them refer patients to you. And also, if you're a site that maybe you've done past trials, reaching out to past patients, obviously within limits, sometimes there's a limit in when they can do the next trial based on a gluten challenge or other drug procedures. So definitely keeping those list of patients and reaching out to them. And then on the flip side, utilizing external recruitment strategies, which I think is becoming super popular right now. Social media campaigns, whether your site has their own social media team, utilizing Facebook advertisements, Instagram advertisements, or if you don't, being able to collaborate with celiac groups, influencers, nonprofits, seeing if they would be willing to promote this study. Our site specifically has gone to celiac expos and tabled and kind of promoted some trials that are going on because that's an event or an environment where a lot of patients with celiac disease might be. And then for sponsor-initiated trials, oftentimes they do have a referral program going on. So definitely making sure that you're signing up for that so that you're kind of covered across the board. Next slide. But in addition to strategies, there's also always pitfalls. So I kind of wanted to touch on some things that sites should be aware of during recruitment phases. A lot of these kind of target more of the external outreach. A lot of external outreach may have a low targeting precision, especially for Facebook ads or Instagram ads. You're kind of reaching a large population, which is what you want. But oftentimes you get patients who are either self-diagnosed, don't have a diagnosis of celiac disease. They live far from the site or states away. You can only put so much on an advertisement, so there is a limited message that gets out there. And sometimes patients aren't fully aware of the trial or study details before marking that they're interested. Um, and then also patients, because of these broader ads, they may have a weaker commitment compared to established patients that you're pulling from your own internal database, which could lead to early dropout. So just making sure that you're kind of being mindful of that. But I also wanted to touch on the fact that using referrals and external strategies could, which is what we want, increase the volume of patients who are reaching out to your site interested in your trial. But making sure that, I guess this is more for sponsors or other sites, if there is a referral program, making sure there is training. I did one study where we were getting referrals daily, but there was a middle person or middle group that was kind of screening them ahead of sending them to the site. But I think there was some gaps in the training of that. So we were just getting a bunch of people that did not have celiac, were not even remotely eligible, which can create a resource burden for staff and CRCs who either they're recruiting at the same time that they're doing patient visits and trying to retain. So just making sure those referral programs are kind of more established. Next slide. So after recruitment, I would say personally, the most important part of a clinical trial is pre-screening. I think it's a critical driver of both successful recruitment, but also retention. And this is kind of your period of you have a patient in front of you. They look like they're going to be a great candidate, but can you now go through kind of things that you can potentially avoid a screen failure for? Um, and so ensuring that the patients are informed, they're engaged, they understand what your study is looking at and the expectations that come with that, making sure that PIs and co-Is, everyone is reviewing these patients that these CRCs are finding and confirming pre-eligibility before screening. Obviously, there are things that are out of our control, lab results, things like that, but you really want to make sure that you are doing the most ahead of time so that you don't have such a high screen failure rate for things that could have been maybe predictable or prevented. And then also pre-screening optimizes your site resources and your timeline. A lot of these sites are multi-site, they're competitive enrollment, and so you don't want to spend time bringing a bunch of patients in for a screening visit, utilizing all your resources only for them to have screen failed because potentially they didn't actually have a confirmed diagnosis of celiac or whatnot. But on the next slide, I'll kind of touch on things that I think should really look at while you're doing your pre-screening checks. Starting with eligibility, I can't stress enough obtaining records of these patients' celiac diagnosis before bringing them in for a screening visit, not relying just on diagnosis, but making sure, especially if the trial requires biopsy-proven celiac, there is obviously a no biopsy approach. So making sure that you are obtaining those biopsy records and confirming that there was MARSH-3 damage. Reviewing key inclusion-exclusion criteria. Of course, making sure that you're very familiar with the inclusion exclusion, but things such as age, a lot of trials specify how long the patient has to be on the gluten-free diet, whether that's 6 months, 12 months, or more, confirming with the patients ahead of time. Any medications that they're on, whether that's prescription, over the counter, and then medical history, because there are a lot of comorbid conditions that are exclusion. So you want to avoid bringing a patient in that potentially has um, type 1 diabetes. I know thyroid oftentimes is allowed if it's well controlled. Um, Another part of this is really getting to know these patients during this pre-screening. So I emphasize this should always be a phone call. Um, and there you want to discuss their symptoms. What are their typical reactions when they're exposed to gluten? And what is their typical compliance with the gluten-free diet? A lot of these things will help you know if they're a good candidate. If you are doing a gluten challenge study that involves 10 grams of gluten for multiple days, and a patient tells you that they vomit immediately with gluten consumption, that's not probably a good patient to be putting in these trials. And then there's also studies that are going on that are looking at patients with ongoing symptoms despite being on a gluten-free diet. And there's normally some variability in there, but you want to really understand maybe current symptoms that they're having, and then again, those gluten reactions. This is also a good time for some feasibility review and making sure these patients understand the expectations of the study. Reviewing the study timeline, procedures. Does this involve endoscopy? Is there a gluten controllant? Is it siege? Blood draws, the placebo component, I think is always really good to stress that these are double-blind placebo-controlled trials. We don't know what they're getting, they don't know what they're getting, and there is a risk that they are getting the placebo. And then also making sure this is, I think, the best time to be identifying those potential barriers with these patients and their availability for these visits. Sometimes there's a visit once a week. So are these patients, do they have reliable transportation? Are they able to get to the hospital? What's their work schedule like? Are they remote? Do they work a 9 to 5? And then do they have any planned vacations or upcoming time off? That's definitely something that I'll touch a little bit on in the next slide, but really making sure that you understand and they understand kind of the commitment that goes into these trials. And then if any CRC is listening to this presentation, I think this is the time where you can figure out retention risk. Once you get off a phone call, you kind of know, you can gauge the motivation, the commitment from these patients. If they're on the phone with you and they're like, this might be a good fit, I'm not sure, they're hesitant. It's completely up to them, obviously. But those are patients that maybe you let them take the time to figure out if they want to do it, but also keep a note of that because that could be a patient that starts the trial and then realizes they don't want to commit anymore. And you can kind of catch that during the pre-screening phase. Next slide. So then going into, you have your patients, you've pre-screened them, you're ready to kind of start the trial with them. It's hard enough to get them, it's harder to retain them. So these are some strategies that I think would be really important to kind of focus on. This first one, reducing participant burden, kind of vague. I also think for sponsors, coming from a protocol design standpoint, one aspect to reducing patient burden would be incorporating home health. I've seen some trials trying to do that. Are there virtual check-ins that you're able to do in lieu of having a patient come all the way into the site, especially for patients that might be traveling far? Traffic, we're located in Boston. Sometimes it takes 30 minutes to go a mile. So just making sure that they're not driving all the way in there if it's a possibility that we can reduce the burden that way from a protocol standpoint. Communication, you'll hear me talk about a lot. Strong communication is key for retaining these patients. No patient wants to join a trial and have limited communication with the study team. They want, so these specifically CRCs and any other study team member, regular check-ins with these patients, sending them reminders, giving them study updates. Check-ins, I think, are also really important for these trials that are going months on end, or if they have months in between each visit. You want to really make sure that you understand how the patient's doing and check in with them. And then focusing on patient experience. These kind of go along with patient burden, but Adequate compensation is crucial for these clinical trials, making sure these patients are being compensated for their travel, meals, their time. As a project manager, I'm very much involved in the contract negotiation and budget startup. And so, first thing I do when I get a budget is I look at that patient compensation line, and that's kind of where we start to negotiate. These patients are taking time off. If it's a gluten challenge, they could be getting really sick and needing more time off. So, just making sure that they're getting adequate compensation. And then making sure that they have easy points of contact for these from the site. Um, Also, kind of looping all of this together, building trust. If the patient sees that you as a CRC is organized, you're in constant communication with them, um, that automatically will build trust. Making sure you're emphasizing our thankfulness for them for giving their time and making sure that they are aware that of the contribution they're giving to advancing our celiac treatments. And then quick turnaround time, this links right back to communication. No patient should be emailing on a Monday and not hearing from anyone on the study team until a Friday. That trust starts with that constant communication. And then get creative. I think as CRCs, that's a lot of this is making sure that you are creating resources or utilizing resources from sponsors if they're provided, creating calendars, handouts, outlines. At screening, we tend to give our patients a folder that in there is any information that they might need, including a 24-hour number, the site contact number, and then outlines of every visit and what is expected at every visit so they can kind of plan ahead. Again, with strategies, there's always a pitfall. So communication gaps. I kind of touched on this already, but infrequent, inconsistent follow-up with your patients or delayed response could make them lose trust or want to drop out of the study if they don't feel that they have any contact with you guys. Lack of reminders or unclear contact workflow. Again, a patient should not be reaching out to you multiple times just for an answer. If you don't have the answer, a quick email back to them, like I'm checking with our study team, we'll get back to you as soon as possible, goes a long way. Expectation misalignment. This really starts during the pre-screening phase, but making sure these patients understand the visit length, the frequency, and never underestimating. It's so much easier to overestimate that than telling a patient the visit should only take 30 minutes and they're stuck there for 2 hours and they didn't plan for that. Um, Make sure there's no surprises around study procedures. Things such as explaining to them the amount of gluten in terms of like how many slices of bread they'll be consuming rather than grams, or how much blood volume they're going to be taking at each visit. And then participant burden. Obviously, there are so many things that are out of our control in clinical research. Wait times happen, cancellations happen, but you don't want that coming from the site to the patient. It happens all the time, patient to site, but making sure that any last minute changes or cancellations are not being done by your site if possible to avoid that. And then obviously within reason, we're pretty limited, at least in our site. Our earliest appointment we can do is at 7 a.m. Or 5 p.m. Obviously, if there is a sample handling or processing that you can't have a patient coming in at 5 p.m. on a Friday. But if you can and you have the flexibility and the protocol allows and you're within window, try and work with the patient's schedule. And then just operational issues that are in control sometimes is just making sure you're not delaying reimbursements or missing invoice submissions or things like that. A patient shouldn't have to email you multiple times just to kind of get their compensation or any reimbursement or travel. So just making sure that you're kind of on top of those things. So, kind of tying this all together, some efficient site workflow is to standardize things, making sure that roles, responsibilities for each staff member are clearly explained, that you're all utilizing a checklist or the same templates, create standard operating procedures or how-tos. But making sure things are as standardized as possible. You don't want each staff member to be working differently. And also, sometimes these trials run years long and you have turnover with CRCs or other staff members. So making sure if it was standardized at the start, it stays that way. Planning ahead. Starting during the pre-screening, our site, we create schedules for patients that literally shows them every visit, the time length, where they need to go, and we give that to them and confirm with them before. Um, Obviously, things happen, life happens. It's so much easier to cancel than it is to try and schedule a last minute endoscopy. I don't even know if that's possible. Next slide. Communicate again. This is more communicating internally and working as a team. So hold weekly meetings with your team, whether that's discussing patients, concerns, issues. Are you having a recruitment holdup? Kind of get together and make sure that everyone's on the same page and kind of brainstorming ways that you guys can kind of fix things. Calendar invites to make sure that patients who are coming in are tracked. The study doctor who is assigned to that patient, that visit, they're aware. Also, in terms of if you're working with the endoscopy unit, you're working with other units in your site, we always send an email to them either the day or a week before, letting them know this patient's coming in next week. This is what we need of you guys. And I think that goes a long way. And then also providing weekly updates to the sponsor. They want to know how your site's doing. They'll check in with you, but it's better if you just start checking in with them weekly. And then document. If it's not documented, it did not happen. Making sure that you're keeping accurate records, you're documenting in real time, whether you have 6 patients that day and you just need to jot something down really quickly, making sure that it's somewhere so that you can then do the final paperwork. But accurate, detailed documentation turns every visit into a roadmap for these safe, smoother future visits. I always like to make sure that you're either taking notes after each visit or documenting things that did happen. I did have one patient, as an example, that came in for an endoscopy. They had an IV placed and had a reaction to the Tegaderm tape. I made a note of it, but then when they came back 2 weeks later for their follow-up endoscopy, I was able to talk to the nurses the day before to make sure they had things supplied so that that did not happen again. So be an advocate for your own patients, making sure that, I mean, that's something she probably would have said anyways, but by the time she got to the endoscopy unit, I had already discussed with the entire endoscopy team so that she was good to go for that procedure. Regulatory, something that is absolutely key for compliance. And so also making sure that if the CRC is in charge of regulatory, you have a regulatory individual who's designated for that. Creating an internal workflow to make sure that one, you're compliant, and two, you're reducing errors. Again, making sure staff are all following the same procedures. Completing case report forms or source documents after each visit or soon after. I know things can get extremely busy, but that data is important and you do not want to wait weeks to be able to be completing that. Um, I would say adverse events and deviations should be absolutely documented in real time, same day, looping in obviously your PI and co-Is as soon as possible. And then ensuring that all scheduling occurs within study window, things like that. And then one thing I would definitely emphasize for regulatory is performing frequent data checks. Don't wait for your scheduled monitor visit. You should be doing this weekly, bi-weekly. If you have someone else who's on protocol, double check your work, things like that. You want to be ready. And then I will end on the investigator site file, which is the backbone of trial documentation for regulatory compliance. So this is a file that either you might have to create your own. A lot of times, if it's sponsor initiated, they are providing you with all of this, but be audit ready. You should be constantly checking this site file, making sure your documents are all up to date, especially as turnover happens and these trials can run years long, making sure staff CVs, good clinical practice, financial disclosure forms are updated. Did you have a change in co-investigators or site units? Then making sure your 1572s are updated. Delegation and training logs, of course. Also emphasizing that training logs should always be updated with protocol amendments. And then always storing your current versions of protocol, consents, IP accountability, and then all correspondences. Everything should be in there. And the more frequently you check it, the less anxiety and stress you have when you have an audit coming up or a monitor visit. So, kind of in summary, every step counts, starting from recruitment, starting from study startup, honestly, when you know that your site is doing a site qualification visit, finding the right patient, outlining the right expectations can lead to the right outcomes, streamlining your workflow causes less burden and better retention, and then accurate documentation, safer audit ready trials, which all go into driving successful clinical trials so that we can hopefully have some more therapies and treatments down the line.
Great. Thank you so much, both of you. Those were excellent presentations. I know we are close to time, but I wanted to ask anyone who's in the audience if they wanted to ask questions. You are welcome to type them in the Q&A and I'll read them, or you're welcome to come off mute and ask the presenters directly.
I'll ask a question. That was a really great presentation. From both of your sites, since sometimes the diagnosis of celiac disease isn't always so clear cut, can you explain how your workflow works when you're screening patients for appropriateness for the trials, starting with just the diagnosis, especially if you're getting outside referrals that aren't in your clinic?
I can start just for referral actually of the patients to the trials. So based on whatever criteria the sponsor gives us, many of them are biopsy proven diagnosis. And so we do require all patients to get all their initial documentation to us from their initial diagnoses. Most of the time that is doable. Sometimes it's really not. Sometimes people are diagnosed in other countries and whatnot. But unfortunately, we just do not refer those patients from trial because God forbid something goes wrong in the trial and they say the numbers, what if you're one number off because you referred a patient who maybe doesn't actually have celiac disease? So I rather not be the cause of that. So I'm very particular about making sure we get every single letter of documentation, not necessarily having to be at our center, but somewhere that I can verify it.
Yeah, and just going off of that, I would say from a CRC standpoint, that's kind of the very first thing we do when we get patients who mark that they're interested or reach out to our site saying that they want to participate. It's kind of the first question we ask of, do you have a diagnosis of celiac and was it biopsy proven? And then we work with them to kind of get those records before kind of moving forward. I know there are some trials that sometimes biopsy proven there's a workaround there, but we always try and stick with making sure we get those records again when possible. Sometimes it's not always possible.
Just to summarize from a workflow standpoint, so the coordinators are helping to get the records, reaching out to the patients first, and then those are getting handed to a physician for review, and that's the flow that both of you guys are using.
Exactly.
Thanks.
Yeah, so we kind of, like if patient's interested, I just tell our research coordinators, this patient is interested, hey, can you just screen them for any trial we have? I might not even be thinking of a specific one. And then they'll go through their history, see what trial they're like possibly can be put into, and then email us back with the documentation we need or whatever it is.
Does anyone else have any questions? Okay, well, if not, I will let everyone run to their next meeting. Thank you so much to both of you for this presentation. Thank you to Takeda for sponsoring this session. And if anyone has any issues claiming CME and needs help with that, you can reach out to us at sscd@celiac.org and we'll get back to you and help you. Okay, thank you.
Thanks, everyone.
Thank you.